Today, Kimberly Jordan will be speaking. She's the assistant research professor in the Department of Immunology and Microbiology, and she is also the assistant director of the Human Immune Monitoring Shared Resource, and she'll be talking about MIBI. So take it away, Kim.
Great. Can you see my screen?
Yep. We see the whole desktop right now.
Yeah, I'm trying to get into, here we go. Okay.
Yep, perfect.
So like Andrew mentioned, I'm going to be talking about MIBI today. Title of my talk is To MIBI or Not to MIBI, Exciting Advancements in High Perimeter Imaging. And I'm hoping that by the end of this discussion, you'll be able to decide whether MIBI is right for your project and the requirements that kind of go into designing a project and what you need to do to get started, really, if you're interested in this technology. And it's not advancing. So I'm the assistant director of the Human Immune Monitoring Shared Resource. I've given a talk to this group in the past, so hopefully you're familiar at least with some of our roles on this campus. We do a lot of pretty much any assay that is related to the immune system in any way. So we start with sample processing for clinical samples. This is really just to help ensure that the samples are cryopreserved correctly for the downstream assays that investigators are interested in doing, namely cell sorting or cytometry. This includes mass and flow cytometry and multispectral cytometry now that we have those instruments available on campus. We also do a lot of immune function assays, so like stimulating cells and seeing what kind of signaling or cytokine molecules they make. Immunoassays to measure multiplex cytokines. But what I'm going to focus on primarily today is our imaging pipeline. So in the HEMSR, we have 3 primary imaging instruments that we use very regularly. We started about 6 years ago with Akoya's Vector 3 platform, and it's still in heavy use, although we've since upgraded to the Vector Polaris as well. So we now have both instruments in our lab. The original Vector 3 can assay 6 markers at a time on a FFPE tissue, so 6 markers plus DAPI. The Akoya Vector Polaris can go up to 8 markers plus DAPI. It also has the advantage that if you are using 6 markers, we can now do whole slide scans that are quantifiable. Whereas with the original Vector 3, we had to use little regions of interest and stamp where we wanted those multispectral images to be collected. And the way they did that was by expanding their filter set. So they have 2 extra filters that allow us to add either 2 extra markers or separate the colors a little better on a 6-color panel, so we don't have to use multispectral imaging and the tricks that that's involved. The Vector Polaris also has a 40x objective. So we've now, in addition to protein assays, we're adding ACD-ish assays to our repertoire. So we can now do imaging of RNA probes as well on the Vector Polaris. The IonPath MIBI scope is the instrument I'm going to spend most of the time talking about today. So it is a high parameter imaging instrument. The resolution is fairly similar to the Vector Polaris, so about 2.25 micron at the highest resolution. It is a rather low throughput instrument compared to the Polaris and does require targeted imaging in regions of interest. So no whole slide scanning on the MIBI scope, but many, many more markers that people can take advantage of, up to 39 actually, currently with the reagent conjugation kits that we have available. So one additional piece of technology that we're thinking about investing in is Akoya's CODEX system. It's fairly similar to the MIBI in that you can get 40 markers on the platform, 40 plus markers on the platform. We're interested in this instrument because eventually the Vector 3 is going to be difficult to maintain its service contract. Most people have switched panels over to the Vector Polaris and are using the advantages of that instrument. Although we are still regularly using the Vector 3 and don't have plans to do this anytime soon, one thought is that in the future, when we're ready to trade in the Vector 3, this may be a potential way of sort of upgrading that instrument to their CODEX platform, which is obviously a lot more markers involved. It's also currently a very low throughput and also very labor intensive instrument. But there's rumors that they're coming out with a new instrument in the near future that may improve that workflow. And so we're interested in sort of partnering with them to see if this is an instrument that our campus would be interested in obtaining. And so we have joined the Akoya STEP program partnership. And what this will allow us to do is serve as the tissue preparation site and liaison to investigators here and some of our biopharma partners, where the tissues are actually sent to Akoya's instrumentation for imaging, but we do the tissue prep and the analysis afterwards. The advantage to our investigators is that they will receive 20% off of all CODEX reagent kits if they participate in this program. And then this also allows us to collect data over the next year or two to see if, again, this would be something sometimes that's not easy to do. With the Akoya vector system, because we're using FFPE tissue primarily, that tissue is inherently very autofluorescent. And so we have to overcome that by using TSA chemistry and amplifying the signal. With IonPath, the antibodies are directly conjugated. There's no secondaries required in most cases. And it's really sort of a one-to-one with the antibody to label. You get more than one antibody obviously going onto a cell, but there's no need for amplification because the background is so low on the IonPath instrument. And then with the vector Polaris, we have obviously a lot more history with this instrument. We've developed over 250 panels across mouse and human tissue, both 7 and 9 color panels. So we just have a lot of experience with these, with this platform, creating custom panels for investigators here. We're really still getting started with the MIBI scope system. And I can, in a few slides, I'll show you the antibodies that we have available currently. You are able to do region selection on the MIBI scope. So we have a secondary electron diagram that we use to visualize the tissue as we're picking regions of interest. Whereas with the vector Polaris, we have an electronic whole slide scan that you can pick regions of interest in. So it's a little bit different, a lot less specific in terms of region selection on the MIBI scope. Okay, so this isn't exactly true, but it seems to be that there's an inverse relationship between throughput and cost and number of parameters that you can obtain information from. So if you think about how do all these technologies work together, I think of it like if you have, a few tissues you want to do a screen on, then these are great technologies to do that because you can get whole genome or whole transcriptome information from your tissue. It's obviously very expensive and a huge number of parameters. Whereas with the vector Polaris, it's a smaller screen with only 9 parameters, but our limitation on the Polaris has nothing to do with staining or imaging. So on our autostainer, we can do batches of 30 slides overnight. We operate our autostainer 7 days a week. With the imaging, we can do approximately 80 slides, and it's more like a 24-hour to 48-hour period. But the bottleneck for Polaris imaging is on the analysis side of things. The MIBI is sort of in between. So it's obviously more expensive than the Polaris platform. The throughput is lower, although with our new commercial instrument upgrade that was just completed this spring, we're now up to about 17 minutes per ROI, where that used to be like 4 hours. So we're in much better shape than we were a year ago in terms of throughput, but compared to the Polaris, it's still a low throughput instrument. So how can you overcome these low throughput issues? For most of the technologies, besides the Visium, you can actually take the approach of creating a tissue microarray. So essentially what this means is you take a bunch of blocks, you core those blocks in specific areas, and you create a single block that has a bunch of different cases embedded in the same block. You can also do what we call a poor man's TMA, which is just putting multiple blocks on a slide, as many as you can fit on the slide within reason. And we can usually get 2 to 3 sections per slide, especially on the MIBI. To create this TMA is an undertaking, to say the least. You need pathologist input or at least guided region selection based on prior staining of each tissue block. The advantage is you can put multiple punches per block, so you're not limited to just like 1 punch per tissue block. You can take 3 or 4 cores from the same block, so you get a representative sample from the block. If you're going to pursue this, we prefer for the MIBI to have 1 millimeter punches or larger, and that's simply so that we can do around 3 ROIs per little TMA spot. And that lets us really sample more of the block. If we have small, really small punches, what ends up happening is it's a square area that has to be imaged, and it's a round punch of tissue. So you get this overlay of really high gold slide coming through, and that just beats at the detector on the instrument, having all that gold being ionized. And so we prefer to stay away from areas that have a lot of gold saturation on the tissue. If you're interested in creating a tissue microarray, you should contact the pathology shared resource for more information on that. So what do you need to get started with the MIBI experiment? The first is tissue preparation. So you have to think about your experiment and how you're going to design it. Carefully select your blocks or TMAs. These sections have to be mounted onto gold slides, and the pathology shared resource is familiar with that process. So all we have to do is give them the gold slides for you, and they can prepare the mounted tissue. Again, we primarily use FFPE, but fresh frozen is an option. Those gold slides then come to Angie in the HIMSR, and we start the staining process. So if you have, if you're just using our antibody backbone that I'll show you here in a couple of slides, then we can just stain with our pre-conjugated antibodies. We can also work on custom antibodies if you have new antibodies that aren't currently on our list. We do have conjugation kits available to make these antibodies compatible with the MIBI system. And we work on new panel development and testing, including staining of experimental slides. Once the slides are stained and dried, they go to Christian in the HIMSR, who is our MIBI engineer and physicist. He has sort of battled with both the beta instrument and our new commercial instrument to really make the workflow happen. So he works on region selection for image acquisition. After the images are acquired, he works on background noise subtraction, getting rid of the gold contamination, for example, and any instrument noise. He's also developed scripts for single cell segmentation and really getting you a single cell staining intensity data spreadsheet that can be used with any other workflow for analyzing those data. So he's both in the image acquisition and in the analysis side of things. And then once you have this spreadsheet, we recommend working with collaborators, statistics and analytic collaborators. We work with a group in the CETA and also in the School of Public Health who can help convert this spatial data into some meaningful data for your biologic condition. And I'll go through some more specifics on that entire workflow as we go through. So our human antibody backbone is fairly well developed. There's really only 2 markers that we're still working on in this backbone, one of which is an ion path antibody that's not working, and one of which is we weren't very happy with our first choice of neutrophil marker, so we're switching over in our backbone to MPO. But essentially the backbone has been established, so we have structural markers that can help give your tissue some context, so lymphatic and blood vessel markers. If you are using a tumor in this case, then there's tumor marker and stroma markers. And then of course, markers that will help us with cell segmentation, where we have nuclear and cytoplasmic markers and membrane markers to help us differentiate the different cell segments. Our phenotyping markers really consist of a lot of immune markers at the moment, and then some functional markers that are also very heavily weighted towards immune cells. We do have a lot of ongoing projects that are going to be adding to this sort of repertoire of antibodies that we're developing. On the mouse side of things, it's a lot more limited. These are the antibodies that IonPath has released and that we can buy off the shelf. So they've separated their antibodies into FFPE and frozen specific antibodies. They don't have the corresponding frozen release for human tissue because it's pretty rare to get fresh frozen human tissue, much more likely that people will have FFPE to work with. On the mouse side of things though, a lot of investigators have frozen tissue. So we have both. We are currently working on expanding these capabilities, and we do that with panel customization. So like I mentioned, all of the antibodies have to be compatible under the same staining conditions. So the very first thing we have to do is make sure, at least for FFPE tissue, that the antibodies work under pH 9 antigen retrieval. And we're going to test this initially on our vector platform, just because we have a lot more familiarity with it and it's a lot easier, faster, cheaper to get it done there. So first is testing the antigen retrieval condition that the antibody works with. And then those antibodies are conjugated to metal ions using the IonPath conjugation kits. We have to test them again pre and post conjugation to make sure that they still work. And we have encountered a few cases where the conjugation itself actually ruins the reactivity of the antibody. So the metal can be deposited on this polymer pretty much anywhere on the antibody, and occasionally we run into antibodies that where the metal gets deposited on the antigen recognition domain. And in that case, the antibodies don't work anymore. So we have to take a secondary approach with those in those cases. And we have run into that a couple of times. Once the antibodies are conjugated, they then get titrated on tissue of interest or control tonsil or both. And those tissues, again, have to be mounted on gold size so we can titrate them on the MIBI. And then we do a full panel test in tissue of interest. So it's a rather long customization process. And it's not fully streamlined yet, but we're getting in our groove as we work through some of these early projects. We are very happy to announce that we are establishing a MIBI antibody reagent bank, very similar to our CyTOF reagent bank that's available in a collaboration between the Cancer Center Flow Core and the HEMSR. So we are sort of taking that model and using it for our MIBI antibody reagent bank. This antibody bank is currently supported by HI3, Dr. Dennis Roop, and the Diabetes Research Center. If anyone else wants to contribute to this antibody bank, please let me know, because these groups will obviously get to have a say in which antibodies we start developing for tissues. And if you're an HI3 member, be expecting to receive a survey very shortly where we're going to be looking to see what antibodies people would like to have in the bank with those funds. I also want to announce that the Cancer Center, if you haven't seen it yet, the Tumor Host Interaction group of the Cancer Center has put together a pilot MIBI project award. These applications are due October 11th. And if you're interested in that, for more information, you can email Jill Slansky. She helped us put together this award to generate some interest in using the MIBI, and they'll be funding 4 applications. And you can use some of the funds from your budget on this award to develop new antibodies as well. And they're really trying to encourage people to develop antibodies that will be useful for across the campus, so that we're not, really project specific, but targets that are going to be beneficial to other Cancer Center members too. Okay, so more on how the MIBI works, and then moving into the analysis of those images and the pipeline that we've developed for analyzing these images. The first is on the image acquisition. So this is an example of the SED image that we see when we're starting to collect, as we're collecting images on the MIBI. So you can find some tissue features. So in gold, I've shown you here, this is a kidney. And you can see some glomeruli. These are these little round structures. It kind of looks like a topographical map of the tissue. And then you can see where the gold is also peeking through these areas. So this is kind of like a grayish area where the gold is coming through. So we can identify regions that have intact tissue as opposed to those that have a lot of gold exposed areas. And it's really, really helpful if we have a similarly oriented H&E stain slide to help us choose these ROIs. So we can use these features as kind of landmarks to help us know where we are in the tissue and to make sure that we're imaging in the right area. And Chris works directly with investigators to choose ROIs as we're collecting images. Once those images are collected, they can be visualized in MIBI Tracker as the IMPass software for visualization. It's a little clunky, but it works to make some pretty pictures for sure. So you can color these kind of however you'd like. They look like IF images or IHC, multiplex IHC images. And in this particular case, this data set was from Ben Bittler's TMA, ovarian cancer TMA data set, where we were looking at patients pre and post treatment with chemotherapy. And the main difference being that we can really see a drop in Ki-67 here, pre compared to post treatment with chemotherapy, which would be expected. The cells are less proliferative. So when we first ventured into MIBI, we knew that we were going to have an analysis problem. The software that we used for our Vectra Polaris pipeline is not open source and can only be used to analyze images that are collected on the Vectra Polaris platform. So being very familiar with that workflow, we were looking for a solution that would give us a similar type of result as what we get from Inform. So we're looking for single cell data where we can, identify macrophages and T cells and B cells and some of the activation markers that those cells express as well. But we really had nothing at the time that we started, and Chris helped really get this pipeline off the ground. This figure was made by Mansouri, by the way. So Chris kind of takes care of the front end here of this pipeline. Mansouri has developed everything inside of the green box, which is more on the phenotyping and clustering side of the workflow. So the first thing that has to happen is mask correction and filtering. That happens sort of online with IMPass software, where the images are recapitulated from the individual pixel data into an image, and those are filtered and gold corrected. So removing all of that contaminating gold that may be peeking through the tissue in certain areas. And then Chris has developed a script for easily looking at the quality of the staining image across multiple images. The problem is that we had a lot of images, and it's very difficult to just click through all of these images to assess staining quality individually. So what we do instead is feed those images into CU-I score is the name of the script that he's developed. And this was really based on a previous script that was available in ImageJ that became outdated and not compatible. So Chris converted this into a macro, and it was originally for an IHC as well, rather than fluorescence data. But we now have both available as a macro that will always be compatible with ImageJ. So that's an advantage, but it's really just based on the scoring intensity. So we look at a histogram of the staining intensity, you split that into 4 quartiles, and then use a weighted ratio to determine its I score on a scale of 0 to 300. And so if we do that across a bunch of images, we can then get that data out very quickly and evaluate whether there are some images that have poor staining quality that need to be evaluated visually. So it kind of narrows down that data set for us. So after we do the quality control, the next step is to segment the image into single cell data. And the problem we encountered during our search for a good segmentation software is inherent to the MIBI images. So these images, if you think about how this process works, the ion gun is ablating each pixel sort of one at a time. That generates a very pixelated image. it's not of the same quality as you get off of immunofluorescence images. You can blur the MIBI images and make them look like fluorescence, but from a raw data perspective, it's very pixelated. And it's difficult for the existing cell segmentation software to deal with because it's not used to looking at these very pixelated images. And especially in areas where we have clumps of cells, it's difficult to tell what belongs to another because you just don't have the same shading and gradients that you have available in IF images. So Chris explored a lot of options, including Cell Profiler and QPath. They have advantages and disadvantages to each of them. And what he ended up coming across was a pixel classifier based approach. So we used the Weka pixel classifier that was already embedded in ImageJ. And what that allows us to do is to classify individual pixels as either being positive or negative for a particular marker, and essentially training it to recognize the markers that we think are positive, or the areas that we think are positive for a particular marker or negative for a particular marker, ahead of doing the cell segmentation. So there was another group out that had been using Elastic upstream of Cell Profiler, which is a similar approach, but he really converted this and made it usable in ImageJ. And again, it's a macro, so it's going to be compatible with future generations of ImageJ as well. So compared to Inform, this is open source, so we can feed our Polaris data into this platform. We can feed any other images that have been generated on any platform into this system, which is really nice. So since he's been working so hard on, on developing this platform, another paper came out very recently that is based on the original DeepCell, machine learning, and it's called Mesmer segmentation. And Mansouri's been working with that software as well, and it does work fairly well for MIBI data. It has really, in order for it to work really well, it needs to have beta tubulin in the panel. So if it doesn't have beta tubulin in the panel, not as good. And also if it hasn't been trained already to recognize those particular tissues that you're using in your image data set, it's not quite as good as when you use an image, a tissue type that has actually been used for its training. So we are continuing to evaluate Mesmer as an option, but in the meantime, we've developed the CU-CellSeg that works really well and is also available. Uh-oh, one of my images didn't come in. Great, well, so I wanted to show you the generation of the probability map that's generated with the tissue class or the pixel classifier. So if you look at this particular signal here from this image, what we're doing is running the pixel classifier ahead of a watershed algorithm to do the cell segmentation. So here you see this like pixelated anti-double strand DNA signal. The probability map that's supposed to be there is really nice and smooth and really normalizes the signal. And you'll actually see that on another slide. I have another example of this probability map. And then the segmentation uses the probability map rather than the raw image data to develop the watershed algorithm to actually generate the nuclear and cytoplasmic and membrane masks that go over the image. So the generation of this probability map solves a few problems. And we noticed that it was solving these problems as we were going through this process of developing the cell segmentation workflow. So it reduces the background noise. It gets rid of like sparse pixels that are sort of in between areas. It helps define individual nuclei and cells, and it normalizes the data onto the same scale. And this actually became really important later on when we were starting to think about the problems we were encountering with phenotyping. So this is the CU cell segmentation that's generated, the map that's generated at the end, and just comparing some of the other options out there for cell segmentation. So we explored Halo, and you can see that Halo generates this sort of cartoonish cobblestone looking segmentation. And I know that there's a lot of options in Halo where you can probably mess with this and get it to be better. But just right off the bat, you can see that there are blank areas where it's calling them cells. It's actually putting an outline on these areas, and it's just filling in all of the space with cells where there should not be cells because there's really no true nuclei there. We think that this segmentation map with CU-CellSeg is a lot more realistic and is better tuned to the actual data that's in the image. Okay, so just comparing, so we took some Polaris images that were developed on the Polaris, imaged on the Polaris, put them through the Inform software that we're using for all of our multiplex IHC image analysis. And we compared it directly to those same images put through CU-CellSeg as a platform. And we can see that we get a very similar number of cells in terms of total counts. We get similar areas for the nuclear, cytoplasmic, and entire cell. We also get very similar staining intensities for various markers that were in this panel. So this tells us that, you know, we're on the right track. At least it's comparable to Inform in terms of analysis, which is really what we were after, is, you know, to generate something that's comparable to our other workflows. So once you finish with a single cell segmentation piece of the data, what is generated is a giant CSV data that we call the raw data table. And this is exported. So this data table contains a bunch of information about the cells, the size and shape of the cells, so various measurements from the cells themselves in different compartments. You get the XY coordinates of each cell and the average pixel intensity of all of the markers across all of the compartments. So this huge CSV file can then be converted into an FCS file and then ran through a number of software. You can, anything that's available for analysis of CyTOF data, for example, or any other large data set, you know, for an FCS file can be used with this particular data. So Chris wrote a script, or just adapted a script to convert the CSV file into a compatible FCS format so that you can easily take the information he's giving you and do your own analysis with Flow software if you're familiar with those options. So Mansouri worked on doing this for our own data sets coming off the MIBI, and we ran into a few problems. As we were working through these, we kind of figured out that there were problems happening over and over. It wasn't specific to a particular project, but was kind of general to MIBI images across the board. So in some cases, we had high background. This is not due to instrument background or lanthanide metals being present in the tissue. This is actually to do with antibody staining. And so because the background is so low on the MIBI, you actually see a lot more background from the cross-reactivity of the antibody or the non-specificity of the antibody or FC binding to these antibodies. So there are high background in some of the channels, particularly in those that have low expression molecules where the signal to noise ratio just isn't real great. There's also a phenomenon of rare bright pixels. So sometimes you get this sort of starry night effect where there's some background. Those rare bright pixels actually contribute to the average pixel intensity that's in the CSV file. And so these were causing problems for the clustering algorithms. And then the third problem is that the data is really not normalized here. So it's really heavily weighted when you do these clustering algorithms towards the markers that are very bright and well expressed. And it's really kind of missing the staining for the low intensity markers. So, you know, as we were working through these problems, we decided to try applying the pixel classification to phenotyping. And Mansouri's really taken this project and run with it on her own. She has a manuscript coming out very soon that we're working on together to just describe this workflow and the algorithm that she's developed. So as you can see here on the left is a raw image of anti-CD3, and on the right is the probability map that's generated ahead of this phenotyping workflow. And if we zoom in a little bit more, you can see these kind of stray pixels that are in between. The cells that are positive are also very pixelated, as we've kind of been talking about this whole time. So the raw data is very different than what you're used to seeing in in traditional multiplex IHC or IF. But the probability map is beautiful. So this probability map takes into account the pixel density in this area, but also, you know, how much other surrounding pixels are also positive, and it generates this probability map. The probability map is then used to export a new CSV file, and this code is called CU-PhenoNorm, and it's located here at this address if you're interested in checking it out. So this new data table contains the same information that the original raw data table contained, but rather than the average pixel intensity with raw data, this is now coming from the probability map instead. So everything's on the same scale from 0 to 100, and it normalizes the data, it gets rid of the background problems, and really makes the clustering algorithms perform much better. So now you can still convert that new data table into a CSV, or from a CSV to an FCS, and use the same clustering tools that are available. So this is a comparison of the same ROIs from the ovarian cancer data set that were collected on the Polaris with a 9-color panel, using Inform on the left and CU-PhenoNorm on the right. And I should mention that Inform is really only as good as the examples you give it, whereas the CU-PhenoNorm approach is really is more unsupervised. So in Inform, you're going through and you're collecting and giving it examples of B cells and macrophages and T cells, and it can only learn the examples that you give it. So there's definitely some inherent error here on the Inform side of things that's dependent on the person training the algorithm. CU-PhenoNorm is also semi-supervised, so there is some you know, level of, when we do the thresholding, we have to make sure that we're setting the right thresholds to say this is positive and this is negative, but it does a better job of clustering some of the cells that, for example, B cells here in green. We actually see a nice little cluster of B cells with the CU-PhenoNorm, whereas they're sort of sporadic and not clustered together, indicating that that's likely an incorrect call on the Inform side of things. So it does actually a better job in some cases of identifying truly positive cells than Inform does. But again, you know, if Inform was trained with a perfect data set, it would likely perform the same. It's just difficult to get that exactly perfect. So I also wanted to show a comparison of when you feed the raw data into some of these cluster algorithms as opposed to using the CU-PhenoNorm. So this is actually on MIBI data now rather than the Polaris data. And we really didn't take this image very much further in getting this perfected to be compared directly to the CU-PhenoNorm because it was so bad. When we're using the raw MIBI values, almost all of the cells are being thrown into this unidentified category, which on the left is shown in blue. And really only the very brightest markers are showing up as something that may be cluster, maybe an actual cell cluster, but most of the cells are being thrown into this negative category. They're not paying any attention. Whereas on the right side, we can see multiple clusters, including a very large tumor cluster. There's definitely still unidentified, which means that those cells didn't stain with any of the markers that happen to be in our panel. But we can definitely see a lot more diversity in the phenotypes that are being identified with CU-PhenoNorm.
Hey, Kim, can I ask a question quick?
Of course.
How are the cell types identified here? Are they manually chosen markers, or is it automated based on like past MIBI data?
No, it's a cluster. So it's cluster. Oh, okay. So all of the clustering is done, and then we go through and look at sort of a histogram of what markers those cells, that group is expressed, so that we look at each cluster and then figure out what markers those are expressing. So yes, Mansouri and I went through and said, okay, this population has CD3 and CD8. That means it's a CD8 T cell. It's not expressing some of the macrophage markers, et cetera, et cetera. So yeah, it was kind of a manual. So to come up with the names for each cluster and what those cell types are is a manual process.
Got it. Thanks.
Okay, so the whole point of doing spatial tissue profiling is to be able to take advantage of the spatial piece of the analysis. Otherwise, it would be so much easier to just do a flow cytometry or mass cytometry experiment if you have access to, you know, fresh tissue that you can break into single cells. Most people that do these kind of experiments are not taking advantage of the spatial analysis. So I really want to encourage, if you're thinking about doing any of these platforms in tissue, that you think about how am I going to use the spatial data. So one thing that Chris is working on at the current moment is getting that CSV file, the giant text file that's exported, to be compatible with Akoya's Phenopter reports that we use for informed data. So it's a really nice open source software that very nicely summarizes all of the data across all of your slides. And it does a rough analysis of spatial analysis. So it gives you, it can calculate an average distance between particular phenotypes, for example, or you can do kind of a rough radius within and say, for example, within every T cell, how many tumor cells or macrophages are within 10 micron or 20 micron or whatever. So it's kind of a rough analysis that can give you an idea of whether it would be worthwhile to pursue a more in-depth spatial analysis. And if you'd like to pursue a more in-depth spatial analysis or start correlating these clinical outcomes, there's a group over in the School of Public Health that Debashis has really kind of developed quite a few postdocs that are interested in these kind of questions, and Julia Robel is working with him as well, and with other collaborators independently to do some more specific spatial analyses. And actually, Ben Bittler and Julia have a paper coming out very soon in the ovarian cancer data set that looks at some spatial analyses there too. So anyway, I highly encourage that if you're going to be thinking about these kind of experiments, to really think how you're going to take advantage of the amount of data that you're generating. What I've seen is that a lot of people are interested in the phenotypes that are there, and they're interested in some of the like markers that are expressed by the cells, but it really sort of stops at that point. And there's so much more data that's being generated that are embedded within these images that, that you could find relationships between, you know, how the cells interact with each other, or how they interact with tumor cells, and take advantage of data that you can't get from other non-spatial platforms. Okay, so I just want to say thank you to my really awesome group of people that work with us in the lab. Jill, our director, Angie, who I kind of showed you where she falls in this workflow. Jen and Jody are primarily our flow and mass cytometry group. Troy is heavily involved in the IHC side of things. He's also working on our ACD-ish our new ACD-ish protocols, and doing some RNA and DNA assays. Our new lab member, Rachel, is primarily working on sample processing, so the rest of the group can do all of these other fun things at the moment, but she'll be transitioning into some IHC work too. And then of course, Chris, as I mentioned throughout the talk, he's our primary MIBI guru and is working on image analysis as well. Just as a shameful plug, we do potentially have an open position coming up soon if anyone knows of anyone that might be interested in working on a BCR sequencing project. This is a new assay for us that we're developing, and it's a sponsored project that's been sponsored for 2 years. We hope to transition that, of course, into a long-term position, but we'll be looking for someone like on a postdoc level, potentially, that might be interested in working on BCR sequencing or learning some of this IHC workflow and analysis that could be useful for their career, or an experienced PRA for that type of position. So that is it, and I can take questions.
Thank you, Kim. That was great. Does anyone have any questions?
Kim, this is Eric Clamby. Great talk. Is it possible in the MIBI to quantify isotopes that are not bound with antibody? Just wondering, yeah, kind of expanding beyond just antibody conjugates to other modalities. Yeah,
so I know there's been reports of RNA probes that are conjugated to a metal ion, but are you talking about like inherent to the tissue kind of
Yeah, you know, for example, I'm just making things up here, but you know, like deuterium, or you know, basically like metabolic studies where you know an animal, for example, was administered an isotope, and I'm just wondering if you could actually track like isotopic distribution within a tissue. Is that possible?
Yeah, so the detector detects everything. So as long as we turn the channel on, it'll detect it. There's no limitation there. So yes, I think that's totally possible. I would be really curious if there is a study like that that would be interested in coming to do some stuff on our instrument. That'd be cool.
Sure. Okay. And there's no lower bound like on CyTOF, right?
No.
Okay. Okay.
Yep.
Great. Thank you.
Yep.
Kim, do you want to drop that link to the Akoya CODEX meeting?
Yes, I do. Sorry, I have to get out of my PowerPoint here. Okay, coming oops to everyone. There's the Zoom link for that. Oh, and it looks like Brad Havarkos had a question. Can you use tissue from an unstained glass slide on the MIBI? The answer is no. The tissue does have to be sectioned directly onto those gold slides in order to be compatible with MIBI. And as of right now, we don't have a way of transferring. If you have a tissue on a glass slide, there's really no good way of transferring that tissue over to the gold slide at the current moment. So it'd have to come off of a block. Awesome.
Okay, any other questions for Kim? All right, well, thank you so much for giving the talk. I thought that was really interesting. I'll be shooting you an email later today about the recording.
Great. And if anyone's in the BBSR is interested in getting involved in analysis of any of these types of images, please let us know. We're always looking for more people that can help with that bottleneck that we currently have in the analysis pipeline.
Sure. Yeah, we should definitely have another meeting.
All right. Thank you all so much.
Thank you, Kim.